Thank you for the introduction. So, as was said, my name is Cassie, and I'm grateful to be here today. And I wanted to thank all of the symposium organizers for the opportunity to speak today and to present my research. So, to begin, I thought I'd give a brief overview of what a crop growth model is for those who are not familiar. A crop growth model is a specific type of simulation model. In my research, I'm using the modeling platform APSIM. Crop growth models bring together weather, crop parameters, and management and allow us to look at nearly hundreds of different trait outputs, including plant growth, crop staging, grain yield, biomass, soil water, and nitrogen cycling, among many others. So I'm using the APSIM Maize model version 7.10, which was released in 2018. And the most important physiological processes in the APSIM Maize model are phenology, biomass production, leaf development and senescence, and biomass partitioning. Phenology is essentially how many growing degree days it takes for the crop to grow from stage to stage, while biomass production is how much carbon is accumulated per simulated day. And essentially the model simulates potential biomass production under water limitations, and biomass production under nitrogen limitations. Biomass production is directly related to leaf development and senescence, as well as biomass partitioning. So my work is on evaluating the use of crop growth models in plant breeding, specifically for simulating hybrid performance and genotype by environment interactions. And what led us to this project was ultimately a lack of work being done to develop comprehensive sets of calibrations that really get at the complexity underlying differences among hybrids. Um, and an approach that many have taken when establishing genetic or cultivar parameters is to just classify hybrids or varieties into maturity groups and assume that genetic parameter values are the same for all hybrids within that maturity. But there are differences among hybrids within a similar maturity for many of the genetic parameters in APSIM, which result in trait variation among the hybrids. And of course, this lack of calibration sets is mainly due to labor constraints associated with obtaining the in-season data needed to derive cultivar-specific parameters. Something that I believe, though, will become less of an issue with advances in in-field, in-season high-throughput phenotyping, but admittedly is a huge limitation when talking at the scale of a breeding program. So also to our knowledge, no other studies have calibrated or generated genetic coefficients, at least to the extent that we have for publicly available XPVP hybrids. And this is important because a lot of information is available on these hybrids, including genetic marker data and pedigree info, which we or others could use to further this research. And lastly, this whole idea of prediction or simulation of G by E holds immense importance and potential for breeding efforts. But most approaches for predicting G by E have been very statistical in nature, and crop growth models provide an added biological or physiological insight, which has been hypothesized to improve prediction accuracies and be interpretive rather than purely descriptive. So the overall objectives of my work have been first to calibrate the APSIM Maize model for 15 XPVP hybrids using empirical field collected data and evaluate what data is needed to obtain adequate calibrations and simulate varietal differences, and then validate or test the calibrations using G2F performance data of these 15 hybrids grown in diverse environments and evaluate whether we are able to simulate the performance and the G by E observed in such trials. So the first step is calibration of the 15 hybrids. We grew out these hybrids in 3 different environments in Ames, Iowa, across 2 years in an RCBD and performed 4 to 5 in-season biomass harvests. So this figure is an example of our calibration efforts. This shows measured field collected data versus simulated outputs from APSIM for one hybrid B73 by Mo17 in 2017 in an Ames, Iowa environment. So the lines indicate simulated values, while the red dots are the measured or the ground truth data collected from the 2017 field experiment throughout the season, along with the corresponding standard errors. And I utilize plots such as these to perform each hybrid calibration manually by first focusing on calibrating for phenology and leaf area, then biomass production, biomass partitioning, grain components, and plant nitrogen. And if we look at model fit for this hybrid in this environment, our calibrations appear to be pretty good with high R-square values and low normalized root mean square errors in general. Overall, our model fit for the calibration portion of this project were good with high R-squared and low normalized root mean square errors in general. And lacking maybe just for traits like stock nitrogen uptake and grain nitrogen concentration. Once we sort of finalized our calibrations to the best of our ability given our 3 environments worth of data, we then utilized the calibrations to run some simulations. So first we used it to better understand the physiological differences among the hybrids for different traits. So we see in these examples just some simulations of the differences among the hybrids for traits like stalk and green leaf biomass throughout the season. And that's where crop growth models ultimately can be very useful to breeders for understanding the physiological differences and traits among hybrids or material, which can be used for selection purposes depending on the trait. Because as plant breeders, say we're selecting on yield in 1 year, and then we select on yield in another year. There's a high probability that you are not selecting on the same underlying trait or physiological trait across years. So for instance, stock biomass is related to stock strength and related to lodging and thus yield. Or another example could be grain nitrogen concentration, where a small change in grain nitrogen concentration can have a large impact on yield. So it's very possible that you could be selecting on some different underlying traits across years, and that's where the knowledge of crop growth models, that what they provide, can be very useful. So to help guide us in the calibration process and help to better understand parameters affecting traits of interest like yield, we performed some sensitivity analyses. And we assessed the sensitivity of 26 different APSIM crop and cultivar input parameters, 17 of which were actually used in our calibrations and are highlighted in blue on the y-axis. So we found that 49% of the yield sensitivity was due to crop development parameters, which really highlights the importance of phenology. 29% was due to crop growth parameters and 22% due to grain component parameters. And it also interestingly shows high sensitivity of parameters like cardinal temperatures, leaf initiation rate, and radiation use efficiency, for example. We're also really interested in using the calibrations to simulate what yield would be like for these 15 hybrids across 20 different years in Ames, Iowa. So we ran a 20-year simulation using an Ames, Iowa environment and historical weather data. And I think this is really interesting because, as shown, we simulate some distinct examples of G by E interactions, specifically crossovers, and among the 15 hybrids, which is really exciting because simulating G by E is ultimately what we hope to be able to do. So I sort of think of this simulation study as sort of a proof of concept. And after performing a simple mixed model analyses of this simulated data, we also found that G by E accounted for 20% of the total variation, which is within typical range. And these plots are from that same analysis of the 20 historical simulated years, but this time in box plot form, with each box plot representing a different hybrid, and looking at 6 different traits: grain yield, grain nitrogen concentration, harvest index, maximum leaf area index, flowering time, and nitrogen surplus. And nitrogen surplus is an indicator for evaluating environmental performance and is just nitrogen fertilizer applied minus grain nitrogen content. And these are just different ways of looking at the data, of course, that can allow us to identify hybrids with high mean performance and low variance, for instance, across environments. But like I said, these are just proof of concept simulations, so the real test is then to utilize these hybrid calibrations to simulate what has been observed and when these hybrids are grown in diverse environments, like has been done in the Genomes to Fields initiative. So for those who are not familiar with Genomes to Fields, it's a public, multi-institutional, collaborative, multi-environment trial, essentially. And the hybrids we've calibrated are grown widely throughout G2F in the majority of the trials. For our purposes, we selected 53 year-by location environments where sites span states like Iowa, Minnesota, Illinois, North Carolina, Delaware, and Texas. and we looked at years 2014 to 2019. So for our 12 hybrids, this gave us a total of 299 observations. If we go back to the same diagram of a crop growth model that I showed before, for this testing portion of the project, we sourced our weather data from NASA Power for all 53 G2F environments. I derived soil profiles for each of the environments using the Web Soil Survey Surgo database. The crop parameters, of course, came from our individual hybrid calibrations, and the management information is supplied by G2F. So using all of these components, we then simulate end-of-season grain yield, and these are what I refer to as our simulated values. The next step then was to evaluate the G2F observed data, which I did using a mixed model analyses to obtain BLUPs and calculate hybrid by location performance estimations. And while these are in theory predictions, I'll refer to these performance estimations as our observed values. So comparing these observed values to our simulated values across all environments and hybrids, we get an R-squared of 0.2, which admittedly is not great, but it's a start with 300 observations, and we're now in the process of investigating and trying to parse out this data to try and understand the random variability at the hybrid by location level where the model isn't predicting well. There are, of course, examples of locations for which we are simulating the hybrid performance quite well. So just one example being for the 12 hybrids grown in the 2016 Crawfordsville, Iowa G2F environment, where we see an R-squared of 0.57. And I'd like to reiterate that these predictions are made using no performance data. It is purely using what environmental data and management data we have, and then the hybrid calibrations. So the first trend we were seeing was that while the model is not always able to simulate specific hybrid by location by year observations well, we were almost always simulating the overall means well. So this bar chart here shows the observed and simulated yield averages for each of the 12 hybrids. And 2 hybrids did stand out as having a larger discrepancy between the observed and simulated means. And you can see where they fall on this scatter plot of the means. It was very interesting to note that these 2 hybrids are also the only 2 hybrids that have LH185 as the male. So there appears to be something about these hybrids that we didn't capture well during the calibration process that may be leading us to not be able to predict its performance very well. Like I said, this is preliminary data and we're still investigating this, but it's an interesting finding and I'm excited to try and understand it. And just as a note, when we remove those 2 hybrids, we are simulating the average hybrid performance at 0.53 R-squared. So if we look at location means for observed and simulated, we again see that we're predicting the overall location means pretty well. But we have some issues in certain environments like North Carolina and one of the Iowa locations, potentially this Iowa 3. To gain a better understanding of why we haven't been able to simulate performance in those environments well, we can start to look at what stresses may have occurred in these environments to impact the yield, and that's something that I'm just now starting to do. Another explanation for the discrepancies between observed and simulated values can come from the uncertainty that's introduced when we don't have complete or accurate management and soil data, which is often the case with these large collaborative efforts like G2F. So to illustrate this, I conducted another sensitivity analysis, this time for just 9 management and soil parameters. And what I'm showing here is just one example of the yield sensitivity, this one being in a Carroll, Iowa environment. And as shown, some of these management and soil parameters can have a large effect on simulated yield performance. And these results really just reinforce what has already been described in the literature, which is the importance of accurate management information and the fact that 10% of the maize yield sensitivity can be attributed to management parameters. So in conclusion then, we were able to develop calibrations in the APSIM maize model for 15 XPVP maize hybrids and simulate our calibration trials quite well with grain yield having an R-squared of 0.89. We found that phenology was very important in developing these calibrations as it accounted for nearly half of the sensitivity and yield and nearly all of the sensitivity in morphological traits like leaf area index. And ultimately, although I can't draw too many conclusions just yet, our prediction accuracies are showing promise, and we're able to predict well the overall hybrid and location means, as well as hybrid performance within certain locations. Um, as would be expected, though, we may not be able to predict performance of the hybrids as well in environments that experience stress, for instance, like a large windstorm that causes lodging and it impacts yield, since the model doesn't account for those things. And again, we highlight the importance of accurate management and soil information to be incorporated in the crop growth model. And just to conclude, I like to always end with the big picture when it comes to crop growth modeling. So ultimately, integration of crop growth models in plant breeding could allow for simulating the performance of material without unnecessary yield trials, a better understanding of your material or your hybrids in terms of potentials and weaknesses for selection. And of course, there's the holy grail, which would be coupling of crop growth models and whole genome prediction. And there have been great landmark papers and studies published on that, as it would lead to prediction of untested genotypes in untested environments. And lastly, more effective placement of cultivar products, as we can use crop growth models to better characterize environments and help with that sort of late stage breeding and product placement. And all of these areas I think are very exciting and warrant more research. And with that, I would like to thank my funding agencies, the graduate students, undergraduates, and staff of the Edwards Lab and Arch and Tooley's Lab, and all of you for listening and providing me the opportunity to present to you today. I'm happy to take any questions. Thanks.